pathogenesis in people in their 70s and 80s. It's a chronic, debilitating disease when its natural history is untreated. And it primarily targets the joints, especially the small synovial joints of the hands and feet. But it's not completely selective in that respect. And there are systemic manifestations, nodules, lung disease, and vasculitis. So as we think about this, and I'm going to be focusing on seropositive rheumatoid arthritis today, but there are really two subsets. There's seropositive people with rheumatoid factor or anti-CCP antibodies, and there's seronegative disease. So we're going to talk a good deal today about seropositive disease because that's where we have the most insight at this time about pathogenesis. And this is just a description of what's going on in the rheumatoid joint, showing the swelling in the digits of the hands. There's inflammation, there's thickening of the synovium, and then there's destruction of the bony joints. This is a view of the inside of a joint, and you can see this is an inflamed joint where we have destruction. And then for years, one of the best tools we've had, we now have ultrasound, but we also have had x-rays. And this just shows the progression of rheumatoid arthritis over time in the joint. And you can see you start seeing a loss of bone formation and then a true erosion. And this is a permanent form of destruction in rheumatoid arthritis that we are trying to avoid in all of our therapies. And then RA is not just a joint disease, and that's going to become more obvious to you as I talk, but it's an inflammatory disease that has multiple sites that are involved. In fact, the life expectancy is 10 years shorter in people with rheumatoid arthritis, likely due not to the joint disabling aspect of the disease, but the inflammation. So we see increased myocardial disease due to atherosclerosis, changes in the liver and fat, bone absorption, and there's even an area of intolerance to stress and depression in patients with RA. A lot of this can be attributed to the production of inflammatory cytokines as part of the disease pathology. So the pathogenesis isn't straightforward, and so this is a kind of classic slide showing the lack of straightforwardness. I'm going to spend most of my time in this world today because I study lymphocytes, and that's where we have the most clues. But when we think about rheumatoid arthritis, we know that we have inflammation that's driven by T cells and B cells, but also the innate immune response. And what we get is inflammation, cytokine production, activation of the innate response with more cytokines that are pro-inflammatory. But we also have B cells that make antibodies. These antibodies form immune complexes, and they also drive further inflammation. At the end of the day, that inflammation is taking place in the joint where there's alterations in the synoviocytes as well as our bone formation, leading to inflammation, pain, and destruction of the joint. So one of the things I thought I'd talk about briefly, I won't spend a lot of time on this, but I wanted to talk about how we treat RA today. We have a lot of rheumatologists in the room who know this as well as I do, but I think it helps for us in medicine to step back and think about what treatments we have, because that helps us think about how are we doing and what are our challenges. And the other important thing for people to know is, of course, in rheumatoid arthritis, we've made incredible advances therapeutically. And so those advances, and sometimes I think, fool people into thinking we're doing okay, but we aren't. So I thought I'd talk about where we're at and then talk about the challenges we face. So some of the drugs we use hydroxychloroquine. I always tell people it's my favorite drug because it's well tolerated. It's not a home run, but it is actually very useful in rheumatoid arthritis and has held up over the years. We now understand its role in altering antigen presentation, toll-like receptor activation, and blocking potentially the interferon pathway. So it's a drug we've used for years, first as an anti-malarial, then we've identified it as useful in autoimmunity, and as we've understand the immune response, we actually now know that it's involved in blocking the inflammatory pathways. Methotrexate's a drug we use. It's the standard of care. Everyone started on that drug, and it's used as a baseline for our clinical trials. We know it inhibits DNA synthesis as a dihydrofolate reductase, but we also know that it has involvement in the adenosine release as an anti-inflammatory. Other drugs we use include leflutamide, azathioprine and sulfasalazine, these are considered disease-modifying agents. But the real kind of breakthrough has been in developing targeted immune therapies, primarily biologic agents. And the first among these are the anti-TNFs that were developed. And this is just showing from a New England Journal article from 2011, the approved drugs that we have for rheumatoid arthritis. So we have the anti-TNFs, these either bind TNF or block the receptor. And these drugs work beautifully, and this is just an example of what it looks like to work. And this is looking at patients being treated with these drugs and then showing their response over time. I want to point out these numbers, ACR 20, 50, and 70. So just think of that as getting 20% better, 50% better, or 70% better. You don't see the word cure here, and you certainly see partial therapy. So I'm going to touch on that a little bit later. But these have been relative breakthrough drugs for us. More recently, tocilizumab, a drug that targets the IL-6 receptor, has become available. And of course, IL-6 is a pro-inflammatory cytokine with impacts across many cell populations and tissues. And so that's been another alternative block for cytokines. Rituximab is a drug that has been used. So that was initially developed to treat lymphomas, but of course targets B cells. It's an antibody that binds to CD20. And then it leads to destruction of B cells and depletion of B cells, which I think to many of us was surprising when we identified it as a way to treat rheumatoid arthritis. So it's actively used in the clinic. And then abatacept is a drug that is CTLA-4 Ig for the immunologists of the room. It actually blocks co-stimulation of CD28 on T cells. So it blocks the activation, we believe, of the pathogenic T cells in RA. And then more recently, JAK inhibitors or small molecules that have come on the market. Tafasitamab is the first one approved. And this, instead of blocking a cytokine on the surface or binding it up, actually blocks the signaling pathway for multiple cytokines and has been shown to be quite useful in the clinic. And the thing about using a small molecule, of course, is that allows us to have an oral therapy instead of an infusion therapy. So we have a big panoply of drugs available in rheumatoid arthritis. So some of the issues that these drugs teach us are raised. Efficacy. When you look at the data for all of these medications, which have different mechanisms of action, that's why I showed you their mechanisms of action, is that we see similar outcomes irrespective of that mechanism of action. So here's a beautiful study where there was a head-to-head trial of abatacept, that's CTLA-4 Ig, and adalimumab, that's an anti-TNF. And in fact, I showed you this data before and didn't explain to you that there's 2 lines for each of these groups. Those are the 2 different drugs. And in fact, in this study that was done head-to-head, they were identical in terms of their response to therapy. I think that's surprising considering the fact that not everyone responds and that these drugs have different mechanisms of action. This is another trial that looks strikingly similar. It's using a not yet available drug, an anti-IL-6, versus adalimumab. And you can see actually that these are superimposable as well. So it raises us some interesting questions for us as we try to select therapy for patients and try to understand how these therapies are actually working, and most importantly, who isn't responding and why aren't they responding and what's the right drug. So the other issue that we face is not only efficacy, but how long does efficacy last in our patients? So we hit a home run with a patient. They come in, they're my 35-year-old, I put her on adalimumab, she comes back a month later, and I am the best thing that there was. It's a great thing to be a rheumatologist. The problem is, 2 years later, she's going to walk in my office, and she's not going to be feeling so good. And that's because she's lost efficacy with her drug. And I am just showing that this data is for tocilizumab. I could have shown you this data for any of the drugs I talked about. So even when we get a response, so as I talked about, less than 50% of our patients actually develop a full clinical response. But only among these, this remission lasts less than a year. And this is why we see this loss of efficacy over time. So that's a challenge we face as rheumatologists. So one of the things we've learned is that we can improve outcome by combining therapies. And we can also change outcome by changing timing. And I'm going to address that in the next slide. So we know that if we add drugs, methotrexate and adalimumab, for example, but this is true for many of our drugs, we actually get an improved outcome. So this is methotrexate alone, adalimumab alone, and this is the combination. And this is actually looking at joint damage by X-ray. So we know we can combine drugs to improve therapy. The other thing we now know is that when we treat people makes a difference. And I think this is really stunning data when we think about it. This is a group of patients who were followed over 10 years. And the difference between these patients is whether they got treated with adalimumab and methotrexate immediately or whether they waited for a year to start the adalimumab. And when you look at these patients, of course you get an earlier response with the earlier therapy. But when you're 10 years out, you continue to see differences. And this is showing the comparator here is this person got a year later, these people got it immediately. And this is at 9 and 10 years, we have a significant difference. And when we look at the X-rays of these people, again, the difference in therapy only goes out to 1 year and then they're treated identically. But at 10 years, the people who had delayed therapy in that first year have increased erosions. So that means how we treat people early really makes a difference. And that's where understanding how RA starts is really important. So how do we, and I think our goal is, I've just told you I have drugs that work great for a while, but I have a chronic debilitating disease. And so the really goal for us is to create a long-term remission or a cure, so to develop tolerance. And where we stand is that, as I said, less than 50% of our patients are able to achieve a clinical remission. And amongst those, that remission lasts less than a year for the majority of them. And now we're starting to move to saying, let's treat people earlier. And we've actually done a series of studies in the rheumatology community, starting very early with aggressive therapy and then stopping drugs at 1 year at clinical remission. And we're able to achieve that long, greater than a year remission off drug in 15% of patients. Now, there's new studies underway that I think are really highlighting how we're going to address these questions using drugs like rituximab, abatacept early. And in fact, Mike Holler's group is starting a trial called Stop RA, where they're going to start treating people before they even develop rheumatoid arthritis. But I'll let him talk about that. So because we have these issues and unmet need, we need to understand where this disease starts, because we're going to have to intervene earlier and we're going to have to do a more targeted form of therapy. And so this is where we are right now, thinking about disease. We have genetic risk. We know there are genes associated, and I'm going to talk a lot today about the HLA class II genes. My group actually studies the PTPN22 gene as well. I'm going to talk about how we define preclinical RA, particularly related to antibodies, and then talk about some of the environmental factors that drive this. So this is the EULAR recommendations and terminology for RA. So we're trying to change our thinking. Instead of people are at risk and then they have disease, we actually have to think about a continuum of people at risk, then developing autoimmunity, and finally clinical disease. So factors that we know are important in RA. So we know the genes are important, and some of the first genes that were described are the HLA class II genes. These are genes that are defining proteins. This is a HLA protein. These proteins present peptides to T cells and activate T cells. Thus, I study T cells. It must be important. And one of the most interesting findings is not that there's one HLA class II gene associated with RA, but there are multiple genes highlighted here, and they share a very common protein sequence that is part of the binding surface of the class II molecule. It's an alpha and beta chain combined, but the beta chain determines these bindings. So we believe that this gene is important in the presentation of protein peptide antigens to T cells, and that could drive disease. Now, the other thing we know is that individuals with rheumatoid arthritis, seropositive rheumatoid arthritis, have antibodies to post-translationally modified proteins. This was discovered while I was a fellow. It really has impacted my research over the years. So, and so when we think about this, this post-translational modification is citrullination. It's actually generated by PAD enzymes, peptidyl arginine deaminase enzymes, which are present in the body, and they take arginines and change them to citrulline. And for the biochemists in the room, you can see that that's a positive charge going to a neutral charge. So it impacts the charge of this protein, it impacts their folding, and we actually, and I'll discuss this more, think it impacts the presentation of those antigens. Citrullination is found ubiquitously, particularly in sites of inflammation, and it has biological roles that we know are important. But anti-CCP antibodies aren't found in people who don't have rheumatoid arthritis. So they're highly specific for RA and 70% sensitive. They recognize these citrulinated peptides from proteins in the synovium, such as fibrinogen, alpha-enolase, and vimentin. These citrulinated proteins are present in the synovial joint, so we know that by looking for them. And their presence and levels correlate with disease severity and outcome. And I'm going to be talking more about this association of citrulinated antibodies to citrulinated proteins and HLA-DR4, which is a very striking association. We know in animal models that T-cells specific for these citrulinated proteins can drive models of arthritis. So one of the most interesting thing is the work that showed that anti-citrulinated antibodies precede the diagnosis of rheumatoid arthritis by an extended period of time. This is from the Denver group. And what they did is they approached the Army-Navy serum repository that contains over 48 million serum samples. These were collected from on-duty military personnel and an average of every 2 years. They weren't collected for this purpose, but they've been repurposed to ask the question, in individuals with rheumatoid arthritis, they had 83 at Walter Reed at the time this study was done, they were able to look at their serum before they developed the diagnosis, and they were able to use a group of healthy matched controls from this cohort. And what they found looking at this, because they had multiple serum samples, that in fact, rheumatoid factor and anti-CCP antibodies precede disease by up to 7 years in these patients. So this is an early indicator that this patient is at high risk for rheumatoid arthritis. Similar findings are true in diabetes. It's been very well characterized for years that development of islet autoantibodies precedes disease, and this is also true in systemic lupus. So it's a marker of autoimmunity before clinical disease. And then with these findings, there's also been raised a new issue is, does RA actually originate in the joint or does it originate elsewhere? And the lung and other mucosal sites have been really the center and focus of this question. We know that rheumatoid arthritis is associated with smoking. I'll show you some of that data. And also silica exposure and pollutants. We know lung disease is associated with RA, and in fact now when we look at early RA patients with high resolution CT, we can see changes. And in fact, when you go into the BAL fluid, you can see anti-citrulline specific antibodies in our RA patients. So it's raised the issue, is actually the initiating factor in these patients not in their joints, but is actually post-translational modification of proteins in an inflamed lung or mucosal surface. Others are very interested in the question of periodontitis, which is increased in patients who have ACPA-positive rheumatoid arthritis. P. gingivalis has abundant endogenous PAD enzymes and drives self and host protein citrullination. We know there's actually ACPA that are crossed from patients with RA that cross-react with P. gingivalis. And these increased titers are associated with disease. So, and there's quite a few studies now looking at periodontitis. A confounder here is that many smokers have periodontitis, so there may be 2 factors there. So we know we have genetics. We know that you develop antibodies early, and we know that there's probably some environmental factors involved there. And then if we follow people over time, we can actually see how the disease develops in the at-risk cohort, and this is data from Stanford, from Jeremy Sokolow's group, looking at the number of autoantibodies to citrulinated epitopes, and these are to multiple antigens, and they increase in frequency and epitope spreading over time. This is actually a heat map showing a cohort of patients as we're looking longitudinally at their antibody and cytokine levels, and these are all healthy controls, and this is 8 years out, and then showing progressively over time, you can see things are heating up as you develop rheumatoid arthritis. So I think that's an important thing to think about. What's our perspective over time? Because if we want to understand the mechanisms that drive this, we need to get back into time. So there's also this question about genes and environment, and there's really beautiful evidence of this link. So anti-CCP antibodies, as I said, are an important marker of seropositive rheumatoid arthritis. And we also know that there is an association with the shared epitope. That's HLA-DR4, 0401, 0404, and 0101. And this is data from Lars Klerskog's group showing that the anti-CCP antibodies that are present are in the patients who have this shared epitope. And when you dive down even further, it's in the shared epitopes that are HLA-DR4. So it's not all rheumatoid arthritis patients who have this. It's actually the rheumatoid arthritis patients who are HLA-DR4, which gives us a much more targeted way to look at the antigens that are important. The other thing that comes out of this group in Sweden as well as others is looking at the environment and its role. And so this is the shared epitope, and this is non-smokers in pink and smokers in green. So in fact, this increased risk of smoking preceding rheumatoid arthritis is very concentrated in the patients who have the shared epitope. If you don't have HLA-DR4, smoking is not a very large risk factor. So there's this interaction between DR4, smoking, and also the development of ACPA. PTPN22, another gene associated with rheumatoid arthritis, also is more important in the patients who have ACPA and have the shared epitope. So there's an interaction between these two genes as well as an interaction with the environment. And this is showing the analysis of looking at smoking and PTPN22. I've highlighted that with the blue arrows. So we have evidence now that there's environmental factors that specifically impact the development of autoantibodies based on HLA class 2. And so that makes us think differently about how rheumatoid arthritis starts and this implication. So this is just a picture of a lung, and we can see this person's a smoker. They have active inflammation, cell death. Pads are activated. There's citrullination of antigens. And then what happens is when I get interested in the story, because we have antigen-presenting cells, HLA-DR4 specific, activating CD4 T cells with these citrulinated antigens, which are now recognized. They're actually foreign to us, but they're recognized, and they by the T cells in the tissue, and then they drive B cell responses and antibody production. And this is a bigger picture of the antigen-presenting cell. This is class 2. This would be the citrulinated peptide, and this would be the autoreactive T cell. This gets activated, and that's actually going to drive B cell development and plasma cell development. So RA may start in the lung, it may start in the mouth, it may start in the gut, and then you develop these anti-citrulline specific responses that lead to joint disease. So trying to understand this link between the class 2 alleles and citrulline and anti-citrulline antibodies has led us to look, us and others, to look at how those peptides would bind in DR4. And you can do this by doing binding profiles where you compare protein peptides that have an arginine or a citrulline. And you can show that actually the binding pockets of class 2, there's 4 binding pockets, preferentially bind a peptide if it's citrulinated. And in fact, when we look at it here, the 0101, 0401, and 0404, these are the shared epitope. They don't bind vimentin in its native form, but if you citrullinate it, they do. This isn't true for other HLA class 2 molecules. So there's this unique feature in the shared epitope of an ability to bind citrulinated peptides. So that gets me to talking about the work that we've been doing in my lab, and we've done collaboratively with quite a few scientists over the years, and that's to address this hypothesis that CD4 T cells that recognize and respond to citrulinated antigens are present in these patients who have CCP-positive rheumatoid arthritis, and that these T cells are actually contributing to the development of these high-affinity antibodies, but probably more importantly, they're what's driving the pathology in rheumatoid arthritis. So that's the hypothesis that we've derived from our observations. And our objective is to be able to visualize these T cells specific for citrulinated epitopes in RA patients with the goal of understanding which proteins and epitopes they recognize. We care about that because these peptides could be targets for antigen-specific tolerance in the future. We also want to know what their frequency and function is, because if we understand unique features of the T cells that are driving rheumatoid arthritis, we could target them better with immunotherapies. And then we want to know if we could detect these prior to disease onset. That's how I'm involved with the SERA cohort. These people who develop autoantibodies 5 years before disease, could we actually see their T cells? And would that help us predict disease? And then we're also interested in understanding the number and function of these T cells in the setting of treatment and disease activity, because it may be a useful tool for understanding response to therapy in our patients before they come in complaining of a recurrent flare. So the approach we've taken is to first develop a relevant panel of tetramers, and I'm going to talk a little bit about tetramers. And so we need to predict the citrullinated peptide that's going to bind in these DR4 alleles. And we do that by assessing their binding to the protein. We then validate this binding as a T cell, as a peptide that can activate a T cell using DR4 transgenic mice. So these are mice that express the human class 2. And then we do in vitro validation with human PBMC. So we show that we have a peptide that binds that can drive a T cell response with immunization. And then we actually show that those T cells are present in patients. But the really important part is we then do something called ex vivo tetramer analysis. And this is where we can take blood directly from patients without manipulation, and we're actually able to count and characterize these T cells in the peripheral blood. And this, I think, has very large implications for our understanding of RA. So predicting citrullinated epitopes, what we do here is we're looking at peptides that in their native situation have arginines, but when they're citrullinated, those arginines become citrulline, and they can then bind class 2. Now, people have worked out over the years the different binding, so they can look at a protein sequence like this, an amino acid sequence, and determine which pieces of that protein can actually bind into class 2. And this is an example of 2 peptides that are derived from fibrinogen, where an arginine is in pocket 4, and it won't bind. But if we change that to a citrulline, it will bind. And so that's been the focus of our work, to identify these unique peptides. We've looked at many different antigens, all of which are known to be in the joint, to be citrullinated in the joint, and be targets of antibodies. So that's how we've picked our proteins. And then we've focused on the class 2 alleles. And this is just a basic demonstration of what this looks like in the laboratory. We take class 2 protein, and then we bind a peptide. We actually do a competitive binding assay. This is showing a flu peptide that binds DR4 beautifully. And we actually compete with that. And if it's vimentin 1, as we've described it here, a non-citrullinated version of vimentin, that doesn't bind or compete. But when we citrullinate that same peptide at position 4, we actually get very nice binding. So this is an approach we use to get our candidates. And with respect to our DR4 work, we've looked at 136 peptides that we thought would be interesting. 24 of those peptides bound. We actually have pairs of peptides where the arginine version doesn't and the citrulline version does. But we also have some peptides where we see both binding. But this has given us an array of peptides to pick from to say, which of these is important? And I want to point out that we're doing this because I don't think there's one peptide that causes rheumatoid arthritis. And I think we have to broaden our look at the antigens and the peptides. And so we've tried to take a broad swath of looking at that. So to validate this, we first use these DR4 transgenic mice. And so we're able to immunize them with the peptide or the protein of interest and then look at their response. So if we immunize with that vimentin 1 that doesn't bind DR4, we actually get no response, and that's shown right here. If we actually immunize with citrullinated vimentin 1, the one that binds well in our binding assay in a plate, we can see that we get a very nice proliferative response from the T cells with that antigen. We don't see any response with the vimentin 1, and we don't see a response with a peptide that binds, but it doesn't activate because we haven't immunized with it. So this is a way for us to validate that it's worth moving forward. Then what we do is we say, okay, now we want to see if we can make a tetramer. And so this is what a tetramer looks like. This is a cartoon. This is HLA class 2. This is a protein that's made actually in insect cells in Bill Kwok's lab at BRI. We then put the peptide of interest into this MHC class 2 molecule, and then we tetramerize the molecule. We actually bind it to streptavidin beads coupled with PE, which is a fluorochrome we use for flow cytometry. So what we do is we take blood cells from DR4 subjects, incubate them with the peptide, grow them up in culture, and then we look to see if this tetramer can be used. And what I want to say is that the tetramer is now going to take the place of the antigen-presenting cell here and actually bind a T cell in a very specific way. And this is showing just a series of the peptides we've looked at, and this is what a tetramer-positive cell is going to look like. It's going to bind the tetramer and be brighter than the other cells. So we use this as a first pass before we move on to the next step, which is ex vivo staining. So there's a challenge here, and that's that antigen-specific T cells in the peripheral blood are rare, particularly CD4 T cells. CD8 T cells against viruses are much more common. Autoantigen-specific T cells are particularly rare. So if we take the blood of a RA patient, or this is actually a healthy patient, HLA-DR4, and we look for flu-specific T cells in this patient, we know they have them, and this is what you find. So these actually, I think, are real. These 2 dots, but that's 2 cells, and we've screened millions of cells to get there. So that's not really a good assay to be using in the lab to characterize rare cells. So what's been developed is an approach where we can enrich with beads for the tetramer-positive cells. They're actually capturing those cells that are linked to tetramer. And then we run them back through the flow cytometer, and now we actually can see these cells. And so these are antigen-specific cells. We're able to actually characterize the frequency of these cells in the peripheral blood based on our initial flow and our enrichment flow. And so in this case, there's 62 flu-specific T cells per million in this subject. And so this has become a tool that allows us to look specifically at antigens. And this is an example then of doing ex vivo staining. Here I'm showing the tetramer bound to the T cell, and that's what we're detecting here. So this is, again, a patient looking at their flu-specific T cells. We use these all the time because they're common and good tetramers. But the important point is because we have multi-parameter flow available to us now, we can use multiple markers. So we can show that these cells are CD45RO, so they're memory cells. They've been previously activated. We can show that they express the co-stimulatory molecule CD28, and importantly, that they express a chemokine receptor CXCR3. This is a Th1 marker for Th1 cells that produce gamma interferon. And so we can go through and better characterize these cells. And then we can go to the more challenging question of using our tetramers for citrullinated antigens. And this is one for a protein called cartilage intermediate layer protein that we've identified peptides to. And here we have fewer cells, but they're detectable. And again, we can see these are memory cells with a mixture of Th1 cells. But I want you to note this number. There's 4 per million in this individual. So we are getting to a point where we need to use 20 million cells to get detectable levels, sometimes even more. Now you can say, great, where's your negative control? And I'm going to tell you negative controls are really hard with tetramers because T cells have a variety of specificities. They're supposed to have that. That's how we respond to new antigens and infections. So it's difficult to get a purely negative control. We have some ways to do that, but I think most importantly, we need to prove the T cells that we're identifying here are actually really specific to the antigen. So what we do is we use these labeled cells. They're still viable in this assay. We sort them individually and do single cell cloning, and then we're actually able to grow up those clones and show that they bind tetramer well and that they actually specifically respond in the context of class 2 to the peptide. This is showing that kill peptide. And so we do this routinely with all the tetramers and antigens we study. So then really the question is, how do we apply this to patients and is it useful? So this is a paper that we published in 2014 just to show you what we were able to do initially with our first set of tetramers. We have 19 RA patients with a broad range of ages, all of whom are DRO401. Also 15 healthy controls, all of whom are DR0401 and are age matched. And then we looked at their frequency. And what we do here is we always look at flu because we need a positive control to know the assay is working. And I think it's actually a really helpful control to detect differences. So the open squares are healthy controls. The dark ones are RA patients. This is response to flu. You can see a nice response in all of our patients, and it's really equivalent in terms of frequency. Then we have each of our separate tetramers here. There's fewer numbers because the ability to test all of the tetramers was not possible in our patients. But when we put it all together, we do see an increased frequency in the tetramer positive cells that are specific to citrulline and RA. It's not hugely convincing, so let me show you a little more data. So this is where actually not just counting the cells helps us, but actually characterizing the cells, because we know healthy people actually have autoreactive T cells in their blood. And so we're seeing them with our tetramers. So it's not a yes or no answer. It's not yes, you have citrulline specific T cells or no. It's really what kind of cells do you have. So if we now look at these cells and we only look at how many are memory cells, so they've been activated in the system, of course all the flu cells are memory, but you can see a much more significant increase in memory cells specific for citrulline, and in fact some of the epitopes start popping out as being significant in themselves. This is actually a better look at that data, so you can see this marked increase in the RA patients as compared to the controls. For citrulline-specific epitopes, there's no difference here with respect to flu. And then if we go down one more layer, which is to look at memory cells that express CXCR3, so these are Th1 memory cells, we can see that this difference gets much larger. So even in the memory population of healthy subjects, they aren't skewed towards this interferon gamma pro-inflammatory T cell. Now, this is interesting from the point of view of trying to understand what T cells are important. In our setting, these are Th1. Other people think Th17 may be important. We aren't seeing them here. The other question you could raise for me is, well, are every tetramer, any cell that binds a tetramer, is it really a Th1 cell? My colleague Eric Lambry at BRI studies allergy, and he has beautiful tetramers looking at peanut-specific T cells. And I can tell you none of them are Th1. They're all Th2. So actually this technology allows us to differentiate these responses in antigen-specific cells, I think, quite nicely. So we were also able, with this relatively small cohort, to look at some other questions. And the first question we looked at was, is there a difference between early onset and later? And we had taken patients across the spectrum, and we had a group of patients who had had less than 5 years of disease and those with more than 5 years. This is our healthy controls, and what's interesting is actually the higher frequency is seen in the less than 5-year patients. And this is true when we look at the CXCR3 as well. Now, importantly, because we think if you're chronically ill, other changes are going on, and we'll address therapy in a second, but the percent of flu cells does not change in the patients, and they're the same as controls. Now, if we also look at another factor that we think is important in our RA is to look at their therapies. Now, all of our patients were on therapy when we recruited them for the study, as most RA patients are, but we divided them into the patients on disease-modifying agents and those on biologics. The majority of the patients on biologics were on anti-TNFs. And we saw this significant difference in terms of frequency with a lower frequency if you were on a biologic. And this was true for Th1 as well. Now, it actually does not correlate with time from diagnosis. So this is independent of time from diagnosis. I asked our statistician to help me with that. And also, when we look at these less than 5-year people, it's actually a much more profound difference here. So we think that we're now able to identify these DR4-specific T cells to citrullinated antigens. And that using these tetramers, we can then describe their frequency and phenotype. Some of the things that come out of this work is that in RA patients, we actually see multiple epitopes specific to citrulline T cells in those patients. So the patients don't have just one T cell, they have multiple specificities. We're now trying to establish a link between these antibodies and T cell responses. Certainly we're looking at the same antigens. We'd like to then go forward to see how the specificity of the antibody compares. We think there's going to be different epitope patterns in different subjects. And as I said, no single epitope is going to be positive in everyone. Patients definitely demonstrated increased frequency of citrulline-specific CD4 T cells, and I think this implies disease relevance. It certainly doesn't prove it, but it implies it. These are predominantly memory cells. They're predominantly Th1 cells. The frequency of these cells is linked to disease duration and therapy, and I think the exciting thing about this is the fact that they're more frequent early in disease may suggest they could be an early indicator of RA development in our at-risk cohorts. So what are the questions that are remaining? Are there other epitope specificities that are important? And we are expanding our search and have identified more autoantigens that we think are likely important. And of course, we've focused on DRO401, but there are other shared epitope alleles we need to think about. One of the questions I'm really interested in is, does the number of epitopes recognized over time or the fine specificity impact disease outcome? In very few minutes, we'll talk a little bit about a multiplex strategy we've created that should address this. And we're also very interested in this question of longitudinal samples, and we're now in the process of creating a longitudinal cohort of patients supported by the DoD to ask this question about change in an individual over time and with therapy. And then I think, we've just scratched the surface with our phenotyping. We can look at up to 12 markers on a cell surface, but are there other important phenotypic attributes? And to do that, I think we need to look at the transcriptome. We're now able to sort these cells, since they're still viable with tetramer, and do RNA-seq either in bulk, or we're able to do individual cells looking at RNA-seq. And then I think this whole question of disease activity, response to therapy is important, and we're addressing that with these. And of course, we'd also like to look in synovial tissue and synovial fluid. We do know these tetramers work in synovial fluid from RA patients through our collaborations with the Karolinska Institute. So I don't want to take too much more time. I just want to talk about some of these technical challenges because I think they may be interesting for people in other fields as well. So, I said that it takes sometimes up to 20 cc's of blood to look at one tetramer. I've told you there's multiple epitopes that we think are recognized, and patients only have so much blood, so we can't really ask all those questions. We do it that way. So we're actually trying to overcome this obstacle where we have a large number of epitopes and we require a high number of cells. And so our ongoing work is to use these tetramers to look at these epitopes and to do the C1, but I'm just going to show you the trick that one of my postdocs has come up with in the lab in collaboration with Eddie James at BRI, which is to miniaturize this assay by using multiplexing. And this is a trick where you can put two different colors on a tetramer, and you can put multiple tetramers in the soup, and you're actually able to multiplicably increase the possibility of seeing these cells. And I'm just going to show you quickly what that allows you to do. It allows you to take 6 epitopes with 4 different colors, and in fact get a very beautiful picture then of a single peptide epitope because we're able to gate out all of these other colors. And this actually shows you from one tube our ability to look at 6 different tetramers. And that allows us to look at 6 different antigens. And this is actually showing single staining versus combinatorial staining. So I'm pretty happy with this. So this allows us to take 20 cc's of blood and interrogate multiple different antigens at the same time. And this is just data looking at people pre and post flu vaccine. And you can actually look at the different responses to flu. You can see that there's a hierarchy. People tend to respond more to the MP97 and less to these more recently introduced epitopes. So it's a tool where we can actually look at the epitopes that are used. This is showing pre and post vaccine. This is one of the new antigens. These are more established antigens in our population. And it can also let you look at cross-reactivity when cells respond to 2 different peptides. So by having new tools, we're able to expand our number of HLA class II tetramers, and I think this is going to allow us to look at frozen cells longitudinally at multiple antigens, both in rheumatoid arthritis, which is my passion, but certainly in response to vaccines or in other autoimmune diseases. And just a last point is that we can profile T cells now in this pipeline where we use the tetramers to characterize them, but Peter Lindsley at BRI is now taking our sorted cells on a C1 machine and actually sequencing individual RNA-seq for these cells. And in doing that, he is able to actually look at the TCR sequence for each cell and show sharing of TCR sequences. This is actually showing type 1 diabetes, where there's increased sharing compared to controls. And this is showing that we can actually group diabetic T cells look different from peanut allergic T cells and peanut tolerant T cells. So we're able to start doing some very exciting transcriptional work with these cells. So to conclude, I think RA continues to be a disease that's challenging to treat and doesn't have a cure. Genetics and environmental factors are involved, and understanding what leads to these factors, I think, are going to help us understand pathogenesis better. CD4 T cells are important in this disease, and we have new tools that are going to allow us to actually dissect their role in disease and what makes them unique in driving RA. And my hope is that using these tools, we can improve our diagnosis, follow response to therapy, and ultimately, I think by identifying these antigens, we're really on the cusp of antigen-specific therapy in rheumatoid arthritis, and I hope that this will help guide that therapy. And just like to acknowledge all the people at BRI, particularly our clinical core, my colleagues at VM who help recruit patients, and my colleagues at the VA, who Bernard is here, who help us recruit for our DoD study. Eddie James Group, my scientists as well, Peter Lindsley, Bill Klock, and of course our funding, and I have great collaborations with Mike and Kevin at Colorado and Lars Klarsbach and Viggy Malmstrom at the Karolinska. So I'll end it there and take any questions. Yes, Gordon.
So, I thought one of the earlier slides was that typical days of peptide to lose the charge. And I have trouble understanding why binding should increase. Is there a repulsive effect?
Right. So an arginine in pocket 4, actually the arginine is charged and it won't fit into pocket 4. So when you get that positive charge goes away, the citrulline will bind. It won't repulse in pocket 4. And there's been quite a bit of work looking at those specific characteristics of class 2.
Yes, Jane. Hi. Beautiful studies, both conceptually and technically. You haven't mentioned regulatory T cells, which I know have been a long interest of yours.
Yeah.
Technically, can you identify Tregs by this technique? And what's your thought about their role now, if you can give us a brief summary?
Yeah. So thank you, Mark, for that question. It is one of my passions. So when we do this phenotyping, we do put CD25 and Win27 on. So we, in theory, can identify Tregs with tetramers. As I showed you, 4 in a million is rare, and Tregs represent 4% of our CD4 T cell population. So I haven't seen them yet, but my colleague Eric, who looks in allergy, where there's a much more robust response, has. So this is one technique. There's some other techniques using activation of cells where we can look at upregulation of CD154 in cultures. And actually CD137, 4-1BB, is used to identify activated Tregs. So that's a new technique that people are using to look at that. So I think we're going to get down the road to be able to look at that soon. Was there another part to that question?
So in the absence of data, what do you think the role is?
Right. So this is interesting, and some people may be familiar with my work. I've studied Tregs for a long time, but one of the things we discovered in type 1 diabetes and then extended in multiple sclerosis is that the effector cells in those diseases are actually resistant to suppression by Tregs. So we think one of the prominent defects in autoimmunity is an inability of the effector cells to be regulated. So I'm actually very interested in the effector population, and one of my questions is, are these cells that we're seeing resistant to suppression by Tregs? And we're trying to explore that more extensively.
Okay, Jane, with regards to those antigen-specific T cells, so one of the drugs, as you know, is Orencia, which blocks T cell antigen-presenting cells. So you might expect that those T cells would disappear. The T cells would disappear quickly after treatment. It takes a while to work. You know, what have you studied?
Yeah, so I can give you just a little high-level view on that. So we are very interested. Right now, we're collecting a cohort of patients Orencia, and then following them at 3, 6, and 1 year. So we'll be able to very specifically talk about the antigen-specific cells. We have recently been working with the Immune Tolerance Network on an MS study that used abatacept. And something I can say is that when you look at patients over time, the level of activation of their T cells is blunted. So if you use CD38, which is about a 7-day out activation marker, you see those percent of cells drop. There's a clear impact on the effector T cells, but we can't wait to see about the tetramers.
Thank you, Dr. Buckner.